Welcome to Medicine Grand Rounds, and it is my pleasure to introduce the Max Mackenzie Grand Rounds in Endocrinology. This is one of the highlights of the conferences in the Division of Endocrinology, and really try to highlight the contributions of Dr. Mackenzie not only to the Division of Endocrinology, but also to thyroid research. Dr. Mackenzie was here at the university for over 30 years. He was chief of endocrine and also chairman of medicine, and he also made extensive contributions to thyroid biology, especially with the pathogenesis of Graves' disease. And this conference highlights his contribution, and is also being funded by the Mackenzie family and a group of friends, and I really appreciate the funding. So I also want to thank Dr. Lou Philipson for accepting the invitation to deliver this lecture, and I'm going to let the chief resident introduce Dr. Philipson.
Hello, everyone. My name is Ryan Severjan. I'm one of the chief residents for the internal medicine program, and it'll be my pleasure today to introduce our speaker, Dr. Philipson. Dr. Philipson is a renowned endocrinologist and a world authority on diabetes. His clinical practice primarily focuses on type 1 and other rare forms of diabetes, and he has a multidisciplinary clinic that treats both adults and children. His approach to care focuses on acknowledging the unique needs of each individual patient. He takes into account their genetics, their family history, and their specific diagnoses. He's also the founding director of the Kovler Diabetes Center and helped establish the first U.S. registry for neonatal and monogenic diabetes with his colleagues Drs. Bell, Greeley, and Naylor. His research career spans over 30 years and began with seminal work on insulin secretion, focusing on ion channels and calcium regulation in pancreatic beta cells. He now leads studies on the genetic causes of diabetes and strategies to help prevent type 1 diabetes. He is also a principal investigator and steering committee member of the NIDDK-funded RADIANT study, which investigates atypical forms of diabetes. And he's published more than 180 peer-reviewed articles, as well as co-edited the American Diabetes Association textbook on atypical diabetes. Throughout his career, he's been deeply involved in the American Diabetes Association. He served as the 2019 president for Science and Medicine, as well as a member of the national board. He currently serves on the steering committee for the ADA EASD Precision Diabetes Medicine Initiative. Finally, Dr. Philipson is the recipient of numerous awards and accolades. Most notably, he received the Banting Medal for Service in 2019 and was also inducted as a laureate of the Lincoln Academy of Illinois in 2018. That is the highest civilian honor in the state of Illinois. Please join me in welcoming Dr. Louis Philipson.
Well, thank you so much. I always warn people that my mother wrote that, but it is a great honor to be here. It gave me a chance actually to read about Dr. Mackenzie, who I think was in the footsteps of our previous dean, Ken Polanski, who when he was starting out as head of endocrinology said that to be a great clinical investigator, what you really want to do is find something and measure it. And by inventing an assay for TSH, basically many years ago, that's exactly what he did. And he also, there was a connection. I had dinner with the father-in-law of a dear friend in Hyde Park just 2 weeks ago, and he is at St. Andrews. And Dr. Mackenzie started his career in St. Andrews and Dundee in Scotland, then went to Montreal, and then came here. So I feel like we've crossed paths a little bit. And then, so and this is conflict of interest disclosure, you have that. There's nothing that I'm going to say, unfortunately, that's directly funded by any of these things. And these are the objectives. So what I'm here to do, and to sort of put the last 25 years in context, is talk about these things. That it turns out that diabetes ought not to be a clinical diagnosis. So if you think that you can look at somebody and think what kind of diabetes they have and what the treatment ought to be, we're not on the same page. And hopefully that's the take-home message today. That's if you fall asleep at this point, that's really what I want to say is you need to make a diagnosis. And we'll discuss how that is and where diabetes should be like other diseases, where there's tools, there's blood tests, there's family history, there's a lot of family history I'll talk about. And those are things that can lead you to make the right diagnosis and to get genetics when you can. But there are other things that will be involved in that. And then there are syndromes. So it's important to sometimes be a lumper and not a splitter, to understand when things actually come together, even though they may seem disparate, that they're actually the same gene expressed in different tissues. And then there are some big wins here, that sometimes if you make the right diagnosis and you say to yourself, why is this person in front of me today? I think Osler might have said something like that. That's a paraphrase. But to understand why is this person with you today? Is it another person with diabetes, or is this the few percent where you're actually going to make a very specific diagnosis that'll change everything for them and potentially for you? So everyone, I think, ought to know that there's basically, we talk about two kinds of diabetes at this point. Historically, they had all sorts of different names. So adult onset or juvenile onset or insulin dependent, which to this day makes me crazy when I see that in a chart and it says IDDM. So I start getting nauseous. But I think that there are things where, then there are miracles. So I send someone to ophthalmology with type 1 and they come back with type 2. So there are things there that are very important to understand. We typically think about type 1, and almost incorrectly, as completely autoimmune. So the autoimmune people have hijacked type 1 diabetes. That's really what they want to talk about. So you measure antibodies. And sometimes the anti-islet antibodies are positive. Sometimes they're not. Sometimes we measure C-peptide, and occasionally that's helpful, but not always. And then lately we found that about half of all cases of type 1 are actually in adults, which is good that we got rid of juvenile diabetes. And then what I point out is that there's something here called a type 1 diabetes genetic risk score, which is not yet ready for prime time altogether, but it's probably the best genetic risk score for any disease in any sort of biological system. It is pretty impressive. And it seems to be reasonably robust across different people of different origins. And type 2 diabetes, we often think about insulin and obesity. That's, again, not always the case. We're talking about lean type 2 diabetes, whatever that is. And there is no single test. So the only thing wrong with these two kinds of diabetes is that we don't know what causes them, we don't know the best way to treat them, and we don't know how to reverse them. Otherwise, we're good. When it comes to monogenic diabetes, we have really a different approach. And of course, there's all the other sort of associated diabetes. If Dr. Weiss was here, he would certainly want me to mention acromegaly and Cushing's, the sort of subtypes of type 2 diabetes, post-pancreatectomy, which looks like type 1 but has a different nomenclature. And then again, measuring C-peptide is not always helpful. It can be, unless it's very high or very low. And otherwise, it's not so good. So the monogenic form, so tomorrow, just to give, for those who want to see part 2, I'm going to talk about mitochondrial diabetes and Wolfram syndrome a little bit more specifically. But for today, I'm going to focus on a little bit of neonatal diabetes, but mostly about monogenic diabetes or maturity onset diabetes of the young, because it really is the poster child for making a diagnosis of diabetes with all the ramifications that means in terms of predicting outcomes, of choosing the right therapy, of understanding then familial diabetes. When people have, everyone in the family tree has diabetes, that you ought to ask yourself why. Of course, that means somebody has to take a family history. It ought not to be the attending. So if you can, medical students, that would be a great thing for them to be sure that they write the family, the pedigree down for all the patients, and you have that to go back to. So I'll talk briefly about forms of neonatal diabetes. That tends to be increasingly important because now these kids are surviving. In fact, I was trying to visit one of my patients who's here in Miami who was born with, we'll talk about a KCNJ11 mutation. And it turns out that while they're eminently treatable, no one wants to take 20 to 30 glyburide pills a day. And we'll talk about that in a minute. We'll talk about the most common, more or less adult forms, but keep in mind that if it's dominantly inherited forms, or even certainly recessive forms, these are lifelong changes in genetic complements of, in this case, insulin secretion, but these are genes that affect many other tissues. So it's a big win if you can connect the dots. So we'll talk more about that. Even though these are particularly not particularly common, and there's arguments about how often all of the monogenic forms together happen. But let's say it's 3%. So that's something like 300 to 500,000 cases in the United States. And if you see in any of the diabetes clinics here, 200 to 1,000 patients a week, you're seeing a whole bunch of these folks. I mean, there's just no doubt. They're not only walking into my clinic. And I think all the cases that I'll talk about today are, in fact, patients that I've seen or people in our clinic. So it can't be that they're just walking into mine. What we're talking about really is precision medicine as applied to diabetes. And there's different ways of talking about precision medicine. You heard in the introduction, I've been involved in some efforts along those lines. But this is the idea that if we can do that, it's a big win in terms of understanding that. And we don't have all the tools yet to do that for diabetes. But what I'm going to talk about are some of the ways that we've done that. Now, as a background, we did mention that I was involved in this international consensus report, which already is a couple of years old. And there's some 14 papers that came out back to back to back over a few months in several different journals, mostly Communications Medicine, but also Lancet and a few other people. And there were 200 different clinicians and translational scientists involved in this effort, led in part by Will Cephalou, who is continued to be the head of NIDDK at NIH. So there's a lot of interest in how to take these tools and apply them to all forms of diabetes, and also then to make specific kinds of sub-endotype, we're calling them now, diagnoses in type 2 diabetes separately. I won't be able to talk about that today very much, but you'll get a sense that there are various applications of this approach to more common forms of diabetes. So what I am going to talk about in the next few minutes are diagnosis, treatment, and prognosis of some specific disease entities related to diabetes. I'm going to talk about our RADIANT grant, which is a 14-center collaboration to identify new kinds of atypical diabetes. I'll mention in just a couple slides our new consortium for monogenic diabetes, and hopefully someone from here or multiple people might be interested in that. And then one slide on precision type 1 diabetes, which involves an interesting gene and also families with multiple generations of autoimmunity as an entirely separate entity. So this is going back to 2006. So before 2006, I saw patients, but I also was a basic scientist, and I was very involved, as you heard, in cloning ion channels. It turns out around 2000, if you cloned anything at all, it was publication quality stuff. And I was very interested in cloning what we thought was the key regulator of insulin secretion. This is the ion channel called the ATP-dependent potassium channel, which turned out to be a superfamily member of a large number of genes related to potassium flux. So I was working on that. My first grants were how to clone the KTP channel. We actually didn't do that. Other people eventually did. But then this young lady walked into my life. And it's one of those situations where we're doctors, we're very arrogant, we think we can change people's lives, but sometimes they change our lives. And because I met this one young lady, that's why I'm really talking to you today. That's how impactful that interaction was. So when we met Lily, she was 6. She was diagnosed with diabetes at 1 month of age, more or less. And exactly how that happened, we don't know. The pediatrician actually wrung out her diaper and measured the sugar. Why did he do that? I mean, it's certainly not standard of care. He must have been burned at some time, but missing something. But this is so rare that it's hard to imagine. So we don't know why he did that, but he did it, and there was sugar in her urine, which is not normal for a neonate. So they called him back, and he decided, well, what could they know? The most common thing is she had very early type 1 diabetes. So she was started on insulin, and if there are anyone who knows infants who've been treated with insulin, it's exceedingly difficult. The pump, is roughly not much smaller than the baby at that point in the older, and certainly 15 years ago or more, almost 20. And so there she was, and there were a couple of episodes where, in fact, she would have seizures related to hypoglycemia and other sorts of issues. Her parents, were obviously hovering that. And then many people with children with type 1 diabetes understand, what the emotional aspect is, but to have it in a baby is also extremely difficult. So around that time, there was a group at Exeter, which still is an amazing group. So Andrew Hattersley and his colleagues were studying genes related to neonatal diabetes and using a sort of a convergent approach, using the notion that some of the mutations cause hyperinsulinism, so the opposite, hypoglycemia, that they were looking at some of those genes and how they merged. And so they gave a talk in 2005, and we heard it just before it was published. And so when we met Lily, and I was face to face with her father at what was then the JDRF board meeting, and he said this, I said, we can figure this out because my colleague Graham Bell is one of the world's leading diabetes geneticists. He's down the hall. Just give us some saliva. We can get the stuff. No IRB, nothing. We just made the oligonucleotides, and within about a week or so, we had determined that she had the same mutation that had been published already, well, it was about to be published in the New England Journal. So we made the diagnosis, and then, and there's a whole story here, I'll show you a little bit more, but at the end of the day, she had a sulfonylurea responsive mutation that could be treated with glyburide, glipizide, glimepiride, all those old-fashioned drugs, and insulin was stopped. We brought her into the hospital. Those days we had to do that, do CRC. Each day we would increase the dose. By day 3, her sugars were higher. Parents thought I was nuts. But by day 7, she was off insulin, and she remains off insulin more or less to this day. It's the more or less that's the problem. And then there was newspaper articles and things, and we thought, okay, well, we'll never see another case. Well, and I'll come back to that in a moment. So it turns out that none of this was new. What was new really was having the genetic tools to do this. But this is, I made this timeline when I was thinking about, as this lecture has developed over the years, really from 1975, Robert Tattersall, who was in Nottingham and probably overlapped with Dr. Mackenzie at different points in their lives, was very interested in inherited forms, and they called it juvenile onset maturity onset diabetes. He went to Michigan and worked with Stephen Fyans in Michigan and started publishing in 1975 that there are families with highly penetrant forms of diabetes, so not new. And also they saw that in terms of some of them didn't need treatment. So many of the things that I'll talk about, we've known now for quite a long time. And then mitochondrial diabetes and deafness came out of Rolf Luft. Some of you know Rolf Luft as being the father of mitochondrial disease, but he was a diabetes investigator at the Karolinska who first published about mitochondrial disease in 1992. And then we have sort of the modern era of Philippe Frugel, who's in France, collecting people from the Paris subway advertisements on glucokinase mutations. I'll talk about that. Dr. Fyans continued now in a, to me, sort of remarkable collaboration with Graham Bell and Ken Polanski and some of the other things. And then my contribution much later, we again came back and rediscovered insulin gene mutations that can cause neonatal diabetes. But you can see the conclusion of this paper from 1975. Our findings provide further evidence for genetic heterogeneity in diabetes, and we suggested in the future it is important that populations in which the genetics of diabetes is to be analyzed should be as clearly defined as possible in terms of phenotype, rather than amalgamated under the umbrella of diabetes. That is my entire message, and that was in 1975. So what was Lily's situation? So she had a mutation in one of the 2 genes that combine to make a potassium channel that's regulated by ATP. And so the basic idea, and these are now sort of classic diagrams that are used all the time in anyone talking about insulin secretion, is that insulin secretion is fundamentally an electrical event. So it's like a neuron or a muscle cell. You need to have activation. You need to have potassium not go through so that calcium can go up. So there's a resting membrane potential. When potassium is blocked, the resting potential can go up. Calcium then goes in, and then a miracle happens. Somehow the insulin granules merge with the cell membrane, and you have insulin secretion. And then there's everything else you might say is commentary. If you have a broken KATP channel, which Lily did, it can be broken in lots of interesting ways. One way is to cause a short circuit. So it's always open. And when potassium is open, it tends to hyperpolarize the cell. So then, and when the cell is hyperpolarized, it can't do its thing and raise calcium. And so then the cell is basically quiescent. Not dead, interestingly enough, but sleeping. So if you add a drug for example, that can block the channel, and the miracle, one of several miracles here, is that glyburide is that drug that blocks the channel. That's how it works in adults as well. Then you get the ability to regulate insulin secretion, and weirdly, according to food as well, because there's a role here for GLP-1, which is now all the rage. So if you eat and GLP-1 goes up, and you have now the drugs that do that, then with food and blocking of this channel by increasing metabolism, you have insulin secretion. So in this case of these children who don't have a functional channel, adding glyburide then can eliminate the need for insulin. And it turns out, though, sadly, there's a huge spectrum of disease. Because the channel is also present in the brain, we have kids who are at different, and then young adults, and now even older adults, now that we recognize them, who are variously impaired. At the severe end of the spectrum, we have this thing called DEN that's been named. So diabetes with encephalopathy or developmental delay encephalopathy and neonatal diabetes. Some of those kids will die in infancy of apnea or seizure disorders. Then there's sort of a whole spectrum depending on how closed the channel is and how responsive it is to sulfonylureas and exactly where that's affecting the brain. So in some of the kids, we have global developmental delay, motor, fine speech, cognition, behavior, interestingly, visual motor sorts of things, and we know exactly which mutations those are. So if we have a child who has neonatal diabetes and we say, okay, you have a methionine instead of a valine in position 59 in the KCNJ11 gene, we know that that child is in for a rocky road. And there are some other then in between that we don't know all the situation. But some of them are actually not so bad. And we have people who've gone on to go to college and have successful lives, and you wouldn't know that there's impairment unless you did really sophisticated neuropsych testing. And then there are some who don't have sort of hardly anything at all, but yet have diabetes. And so there's still a marginal effect on the insulin secretion. And then there's even a few mutations that have been linked to whatever type 2 diabetes is, although typically they tend to be non-obese. So that was a little bit about that. And the family was really so taken with the whole thing, and they had some means that they made a movie. So if visual learning is your thing, you can find this movie on YouTube and Vimeo and whatever your favorite streaming service is. And it's already 10 years out. So I'm a talking head, and you see I look entirely different. And there's pictures of Dr. Hattersley sort of striding across the moor in southern England, and Fran Ashcroft, and it's now Sir Andrew and Dame Ashcroft because they've been recognized for their work in the UK. So again, this was something prominent because of the family's interest. Really, as I say, the lives changed, and I'll talk about that, how we then started making registries for not only neonatal diabetes, but for adult onset or adult recognized monogenic diabetes. And as I said, it's not that rare. So something like 3%, maybe more of all diabetes, usually misdiagnosed, usually dominant inheritance, but there are sadly recessive forms that can be even more severe. But when you can pick one of these things up, it's, as I said, it's a very interesting thing. It can be a huge win. And then there are, I'm speaking mostly about mutations that affect insulin secretion, but then there's a whole host of mutations we won't have time to discuss today that are on the other end of the spectrum. Mutations that affect insulin signaling defects, which could be, for example, at the level of the insulin receptor. So those are severe and well characterized, with also mental retardation and short stature and early death. Lipodystrophy syndrome, some of those involve insulin signaling, and now ciliopathy-related diabetes and insulin resistance. So there's a lot of stuff here on both ends of the spectrum. I'll mention at the end that, I mean, this is sort of the overlap in genes. So this is already a few years ago, and this, well, actually 10 almost, from Larry Chan, who was chairman at Baylor at that time of endocrinology. And so they put this together just to show even then this overlap between genes associated with type 2 diabetes, genes associated with type 1 diabetes, autoimmunity, and then monogenic diabetes, and then overlaps. So there are interesting sorts of ways that the immune system, insulin secretion, metabolism, regulation plays a role in how we're understanding this a little bit more. So now let's focus in on a few of the specific cases that we've seen over the years. So as I said, this really started with, in this country, with Steve Fyans and Robert Tattersall, who came briefly to Michigan and helped Dr. Fyans get going. And then the big change was when Dr. Fyans had started a collaboration with Graham Bell. And if any of you are interested in the genetics of diabetes, you'll see there's a lot of papers with GI Bell, Graham Ian Bell. And so it's hard to understand that it's like one person because there are so many people, there were so many papers. There was a time between 1995 and 2003 where I think it seemed like there was one Nature or Science or Cell paper a week from the group. A whole bunch of postdocs who like never left the lab. There were mattresses and food, and I'm not kidding. I mean, there were mattresses in the lab. So that was what happened. And then they teamed together. Graham had already cloned the insulin gene, the insulin receptor gene, glucagon, somatostatin genes, opioid genes, and that's like the beginning of the list. So he got very interested in this aspect because there was hope that understanding these genes would tell us a lot about whatever diabetes really is. So this was one of the families that Fyans had put together. He called it the RW pedigree. You can see the Roman numerals, those are generations. So there's 6 generations here. The black circles and squares are people with diabetes. And they started writing about them way back in 2017. Simon Heller, who is also a famous diabetologist in the UK, wrote a life, kind of a bio of Tattersall the year before he passed away. And there were several other things about Tattersall that were interesting. Some of you may have heard there's a phrase called dead in bed, of children especially who have type 1 who don't wake up. And that was one of Tattersall's contributions, looking at the excess mortality in children on insulin with type 1 diabetes. And there were some other great things, including also diabetes, euglycemic ketoacidosis was seen at that time as well. So what Graham did was to start doing brute force cloning at that time. So there wasn't really a lot of roadmaps. So you had to kind of figure out what chromosome a mutation might be in, and this was what Graham would call an informative family. So by putting all that together, getting DNA from everybody, sifting through where the chromosome was, and then using lots of postdocs to find out that, in fact, there was a mutation in a gene called HNF4 alpha, which was a big surprise because HNF stands for hepatic nuclear factor, and it had already been cloned. Just no one knew it had anything to do with insulin secretion. It was in a liver library, a DNA library, cDNA library, and it didn't seem to have too much in the liver. So it was sort of a disappointment. It turned out to have a whole lot to do with insulin and islet development. So this is with Lily being case 1, here's this case 2. So this is one of the people who we saw. She was 58 when we first saw her. She had diabetes at age 19 with a mild elevated blood sugar, and there's this confluence of numbers. She was 19 years old with a BMI of 19, so not typical for type 2 diabetes. Then she became pregnant, and that was confusing. Couldn't really be gestational because she already had pre-existing diabetes. Was it type 2? Was it something else? And then over the years, she had all these fluctuating things. By the time she saw the new endocrinologist, not me, but someone in the South, she was 58. She had moderate insulin resistance. So, our normal ballpark is about half a unit of insulin per kilo per day, is sort of being reasonably normally insulin sensitive. So she was 2x at 90 units per kilo per day on a pump, but she gained 100 pounds. So she was now 230 pounds with a BMI of 40. That happens when you're on insulin because you're terrified of having a low blood sugar. And so insulin doesn't cause the weight gain. It's the terror of hypoglycemia, which generally speaking causes weight gain along with insulin. So when he took a family history, he saw this. So the orange arrow points to our patient. Her 2 sons had diabetes, and then, my goodness, so did her mother, her aunts, her uncles, her grandmother, and a whole lot of other people. There are some red herrings here of people who were probably not the same mutation, but many of them were. And the 2 sons, interestingly enough, were very proud of themselves being type 1 diabetes. They were very active in that community. But what Graham had figured out was, because we knew exactly where the mutation was in HNF1 alpha, that in fact all of these people who agreed to be studied had the same mutation. So this was our first understanding that not everybody wants to be tested. So people sometimes just are happy the way they are, and there's inertia, both therapeutically and diagnostically, but also from the interaction. And if the doctor says that's ridiculous, there's no such thing as monogenic diabetes, as happens to this day, then there's a difficulty in making a diagnosis. So all of this was important. And then again, this is the same tree. That's the mutation. So it was a stop codon at position 352. So a serine became a proline, and that led to a stop. So there were a few takeaways before I go into more details. One is that just because you're overweight doesn't mean we know what you have. You can have type 1 or type 2 or something interesting like this, something more interesting potentially. Family history is essentially everything. So I might say that a dozen more times, and so maybe some of you will remember that. Take a family history. And then in this case, the big win is that HNF1 alpha, which is also called MODY3, we're trying to get away from the numbers. That's like not helpful. I don't remember what MODY14 is. So we try to say, what is the gene? And that really clarifies the situation. So HNF1 alpha is remarkable in that it responds extremely well to sulfonylureas. So well, in fact, that some of these folks have hypoglycemia to the first dose. So there are two reactions to that as a clinician. One would be, oh, I'm not going to use that drug again. And the other was, gee, that's interesting. How come they're hypoglycemic? And maybe we could go with half the dose or a quarter or a tenth of the dose, and it would still work. And so these people tend to be hyperresponsive to sulfonylureas, at least in the beginning. So this is what happened. She started sulfonylureas. Her A1C improved into the 6% range. Insulin withdrawn. She lost weight. In fact, she's lost, I mean, I'm still in touch with her. This was a few years ago. She's lost close to 90 pounds. And then again, so there was an ambivalence about the children and some of the family members because their identity was so much embedded in the kind of diabetes they had. So it took a while before they saw the wisdom of that, and especially when they understood dominant inheritance, and both her sons had children. So they understood that it was important to be able to understand how best to treat their own children. And then weirdly, one of her other doctors thought, gee, she has diabetes, let's try an SGLT2 inhibitor. So that was with her days when SGLT2 inhibitors came out. It seemed like a good idea. Wasn't a good idea. So the people that you ought not to give SGLT2 inhibitors, and there's 3 of them on the market at least, maybe 4, are those people who are not making a lot of insulin. So I will talk about that in a moment. I'll come back to that in a second. You can think about that for now. But this is what the transcription, this is a linear cartoon of this particular transcription factor. So that's what HNF1 alpha is. It is a control software situation for lots of downstream effects. It sits on the DNA and interacts with the proteins to turn other genes on and off. And this just gives you a sense of the specific domains of the protein. So can you see my? Yeah, so you have in the amino terminal a dimerization domain. So you have at least 2 of these things have to bind to each other. Then there's these other other sorts of key domains for DNA binding, and then a transactivation domain to bind to other proteins that are also sitting on the DNA. On the bottom are a handful of mutations that we illustrated there, and this was done by Jose Flores, who's now chairman of medicine at Mass General. But this is what a map looks like of most of the mutations. Now, this map was old. We had a medical student do this in our group a few years ago. This was the beginning. So these are all the mutations. So all the red ones, most of the red ones are mutations that cause monogenic diabetes. There's some that are benign and some that are called VUSs. So it's always a head scratcher, variants of uncertain significance. And these are just point mutations. What's not here are the mutations that are deletions, so large sections of the gene can be eliminated. So what it does tell you is that it's a miracle that anybody gets up in the morning. I mean, this is the mutations in one gene, and all the genes are like this. So it gives you a sense of the power of the technique, and also how amazing it is that the variants can be identified. Our lady had her mutation right here where this figure is, and it meant that she had a stop codon there, and so the protein probably was never made. So there's different levels of mutation. Sometimes the protein is made and doesn't work. Sometimes the RNA is unstable and so never gets translated. We think that's the case over here. So I mentioned the euglycemic DKA. So one of the really interesting things as a transcription factor is that HNF1 alpha controls the expression of the protein SGLT2. So for the cardiologists, you know, it's all the rage, right? Everybody should get an SGLT2 inhibitor, at least that's what we thought at one point, and it affects all sorts of good things. But if you don't have much in the way of insulin secretion, you can then have a situation where your renal output of glucose makes for insufficient insulin function. So you have two effects of insulin. So there's insulin which causes your blood sugar to go down, and there's insulin which turns off your ketone production. Two completely separate mechanisms. So if you don't have enough, so you could have a normal blood sugar, but insufficient insulin to suppress ketone formation, which is actually also a transcriptional event, then you're going to have ketones even with a normal blood sugar. And Our Lady, with a well-meaning physician, gave her one of these drugs, and she was in the ICU for 3 days with severe DKA until, you know, that was figured out. And this was known in 2000 in studies, so 25 years ago, in studies with mice. So again, just to summarize that, because the HNF1 alpha is a transcription factor regulating SGLT2 expression in the kidney, there's already a decrease in SGLT2 expression if you have 50% of HNF1 alpha. Then if you further block SGLT2 with an SGLT2 inhibitor, now you have a flood of glucose coming out into the urine. So, and then insulin goes down or insulin requirements go down. It's a miracle, but the problem is ketones. So that's a little bit about ramifications of that. And then there's a related gene, HNF4 alpha, which was also found. I won't go into the family and the details so much there. But there are some other things that fall out of this. One is that there seems to be an increase in various subtypes of cholesterol, including LP-little-a. So that has to do with the otherwise perhaps minor effects of HNF4 alpha and 1 alpha on the liver itself, and the fact that they also can regulate various apolipoproteins in modest ways. If you also can have neonatal hypoglycemia, not well understood. So these are, again, the children of people with one of these mutations who then the child will have the same thing, but then there is a high birth weight, possibly because of mom having diabetes and having to need treatment. So the babies can have neonatal hypoglycemia, and that needs to be thought about. Liver adenomas can happen, and that's interesting because HNF1 alpha is in the pathway for hepatic carcinoma, so hepatic adenocarcinoma. So the genes that can happen there include mutations in HNF1 alpha. So we do find that in a small number of these folks, there are liver adenomas. We don't ever see cancer, at least not yet, but we are seeing some cases, large adenomas which require surgery. And then also, and to some extent, surprisingly, hsCRP is very low. And that can be an indicator of inflammation. So we use that in various conditions, diabetes, obesity, or inflammatory conditions and infectious diseases. But the expression of the C-reactive protein is also in part controlled by HNF1 alpha through the liver. So these people have low hsCRP, and whether that's a cause of anything or simply a marker, you know, we're not sure yet. Another case, so this is diabetes or hyperglycemia, and this is one of my favorites. So again, a young man, this is a family history, young man with elevated blood sugars, his sister, father, an aunt and an uncle. Fasting blood sugar is mildly elevated, A1C just in this case below the range of diabetes. Those of you who know A1C is the cutoff 6.5 for having diabetes, but 5.7 is normal. So you have this in-between area where, you know, it could go either way. But again, BMI is 23. So what is that all about with someone who is actually left shifted from normal? Antibody tests for type 1 were negative. And by the time we saw him, he had diabetes insulin-treated diabetes for 13 years. But someone said, okay, we need to think about genetic causes of diabetes, and then that revealed a mutation in glucokinase. And then this again is the larger family tree. And what about glucokinase? So this figure was from 2001. It's still very effective, and it was a review article in Science from my teachers, Graham Bell and Ken Polanski. And you can see they tried to put everything together. So there's an insulin receptor, there's a calcium channel, there's the KATP channel, and then on the left is a glucose transporter, which is a whole different topic. But the glucokinase happens here. So without glucokinase, glucose doesn't get trapped in the cell and you don't have metabolism. So what is very exciting and interesting about glucokinase is that it is one of the regulators of insulin secretion, but also of glycogenolysis in the liver. And we also know that glucokinase is expressed in many areas of the brain, those three places. And I'll come back to the brain story in a moment. So people who have half the complement of glucokinase are not able to regulate insulin secretion normally. And I'll come back to the sleep disturbance and such. So here you see on the left is just a linear diagram with all the amino acid letters of just a linear glucokinase, a mutation map again from a few years ago. It is very interesting that because glucokinase folds up being an enzyme and not only a transcription factor, it has to fold up in a very particular way. So the original, there's this called the butterfly hypothesis here. So you have a binding site for ATP and glucose right in the middle. And then when ATP and glucose bind, then this another miracle happens, the two wings close, and you get a transfer of phosphate from ATP to glucose. And we have glucose 1 and glucose 6-phosphate happening. So that active site is all folded up. And so there's not a linear relationship necessarily between where that active site is and the linear map of the protein. And then also you can see there's some issues way out at the amino terminal and the carboxyl terminal, which couldn't possibly have anything to do with the active site, but may have to do with binding, because there's evidence, we were talking about David Piston at WashU as some of this evidence that glucokinase migrates in the cell and binds to mitochondria as well. And so some of these other things that cause defects in glucokinase function could also involve secondary binding activity. So why does it work? It's because glucokinase is tuned, if you will, to best reflect blood sugar, so that's an amazing thing. So you can see normal wild-type glucokinase in the green line has a half maximal right around normal blood sugar, right around 90 milligrams per deciliter in this linear range. If you take away 50% of the glucokinase, or you put in some mutations that can affect binding or KM or Vmax, then you have this, what we're calling MODY2, or glucokinase-related hyperglycemia. And if you have two mutations, you have essentially no insulin secretion at all. So there are rarely babies born, which unfortunately get one bad glucokinase from each parent, and then bad things happen and they need insulin from life. You don't come back from that. The payoff here is that glucokinase mutations are modest in terms of their effectiveness and lifelong effect. And so there's huge impact here, I think, for understanding how to treat diabetes altogether. So in this particular case, this is an early slide it was a slide early on in the use of continuous glucose monitoring. And this patient, interestingly enough, was a rocket scientist, and he felt that he didn't need insulin. So he left his CGM on with and without his insulin pump. And as you can see, there was really no difference. His doctors were surprised because they had really asked him not to take off his insulin pump. But because of the balanced mutation in the insulin secretion and in the liver production of glucose from glycogen, both things are working in opposite directions, there's not much you can do. You need a boatload of insulin to really reduce the blood sugar. It's a particular problem, for example, in pregnancy. And so this can be a whole other issue, which not time to talk about today. But meanwhile, the main thing here is that no treatment at all is what's required. So this is a big win. So if you find someone who's on insulin or lots of drugs and they're trying to diet and exercise, our colleague at Exeter, Dr. Hattersley, says that's ridiculous. They require no treatment at all, not even advice on diet and exercise. Just do what you would do otherwise, because it's not going to affect anything. And in fact, people in some of the large collections, because this is one of the most common mutations, 80, 90 years of age, maybe there's some dot blot hemorrhages in the eye from complications of diabetes, but otherwise not much. And as you can see, what this is the line of A1C going up with age. Even people without diabetes have a rising age dependence to A1C, and people with diabetes, a little bit more. So I'm just going to go through a couple more things and then hopefully leave time for questions. So misdiagnosed, improperly treated, it isn't pre-diabetes, it's modestly or non-progressive hyperglycemia, which doesn't need to be treated at all. And we think about 300,000 people in the United States alone, of which we know maybe 2%. So if you find anybody in your clinic, you'll increase that number dramatically. And I'll tell you, there's another aspect too. So I mentioned that there's a sleep dependence. So we have a wonderful former fellow who's now in San Antonio, who originally went to medical school in Ecuador and spent time here as a resident in Miami at this place. So this is Marilyn Orozamina, and she's a remarkable, dedicated clinical scientist in addition to being a wonderful clinician. So she's published 3 papers with us on sleep and monogenic diabetes. So this was the first one in 2023. We were interested because we have also a very effective sleep center. We know about sleep and type 2 diabetes and obstructive sleep apnea and other sorts of defects in sleep architecture. But we wanted to know, well, what if you took away the insulin resistance and the metabolic issues in type 2 diabetes and looked mostly at pure hyperglycemia? So to do that, we took people from our registry had either a transcription factor diabetes, what I mentioned, HNF1 alpha or 4 alpha, or glucokinase, and we compared them in various ways. And what was remarkable is that the glucokinase folks had essentially normal sleep architecture, whereas the HNF people were in between sort of normal and severe diabetes, obesity-related sleep disease. And then more recently, this paper is about to come out. We then looked harder at the literature, and you can just maybe just on the conclusion that these folks had a lower resting heart rate, which we're not sure what that's all about. It could be that because glucokinase is in the brain and some of those areas in the brain affect the vagus, and we know that resting heart rate is a correlate of cardiovascular disease. The higher your resting heart rate, that correlates with mortality. So it's not just an observation. So the fact that they have lower resting heart rate may relate to the fact that they do well over time with their hyperglycemia. So hyperglycemia alone is not a problem if it's not too extensive. So I think there's some interesting messages here to take home. I'll do one more and then I'm going to try to wrap up. The T pedigree I'll just mention very briefly. So when we met that young lady I showed you in the beginning, other people started calling us. And again, I thought I'd never see another case. But this lady called us and this was her family history. And again, having Dr. Bell in the building meant that we could instantly look for all her genes. She mapped this mutation mapped to chromosome 11. He immediately had been thinking about insulin. Well, we all thought about insulin because of Don Steiner being another one of my teachers who discovered proinsulin. So it turned out that this was a mutation in the insulin gene itself as a cause of neonatal diabetes. That really hadn't been described, but Dr. Steiner was interested in evolutionary aspects of diabetes, and so this is a map showing that region of insulin gene from humans to cattle, sheep, horse, whale, rattlesnake, and then IGFs and so on. And in that position, which is called the 8th position in the B chain, everybody has a glycine. And I mean, like, evolutionarily speaking, all creatures have, if they have insulin, they have a glycine, with some exceptions, catfish, IGF. So this turns out to be a hugely important thing. And then insulin is really so interesting in terms of where it is, right? It's only, for the most part, in insulin-secreting cells in the pancreas, maybe thymus a little bit more, 20% of the total message, 10% of the total protein. And by its very nature, it induces ER stress because every time you have one molecule of insulin, you have 3 disulfides that have to merge, and each disulfide generates an oxygen radical. So the more insulin you need, the more damage you're making to those insulin-secreting cells. And this is a theoretical picture of what happens when that B8 glycine on the left becomes a serine on the right. It sort of flips everything out. And we don't think this ever gets made, but if it did get made, you wouldn't certainly have a functional insulin. So every diabetologist, I think, dreams of showing a structure of insulin in a lecture, maybe. And so this is, a map that we started making more than 15 years ago. And again, so not all the mutations, but all the black ones cause neonatal diabetes. All the green ones are interesting lab curiosities of hyperproinsulinemia. That's because there's a defect in insulin processing, which really doesn't have much effect on blood sugar, but creates a strange, the people scratch their heads, what did you send them? And then there are some that are purple. So there's one on the bottom right. That purple one causes adult onset diabetes. So there's some slow destruction of the insulin secreting cells in minor abnormalities. And you can see Our Lady, who had the glycine here, changed to serine, that turns out to be a hotspot. So there are multiple mutations there. And then even some mutations in the leader sequence, which this is in actually pre-proinsulin. And there are mutations way out here which also can cause diabetes that are not coding. So this turned out to be very interesting. That paper from, and the other, it's had over 500 citations so far. And then this is the end of this part of the lecture, and I'll see if I can wrap up in 5 minutes. You can see here MODY 1 to 14, which is not helpful. So we don't use that designation anymore. But I think if you know a few of them, you know that there is HNF1 alpha, glucokinase, really that's important. The map tells us how we think about these mutations, gene expression, ER stress. There are some mutations that affect the exocrine pancreas. So you might say cystic fibrosis is in that category, maybe hemochromatosis. And then I just want to talk about a few more things. Could be another hour, but I won't do that. So our goals are to continue to identify new cases, to educate, understand what return of genetic results is all about. That turns out to be quite confusing. Most doctors don't get genetic testing results clearly. And cascade testing, I'll show you in a moment. And then to build a data commons for the community is really what we're doing. So right now we have something like 4,500 people in our registry over the last 15, 20 years. And you can see over 1,500 of them have a known cause. So some of the others are type 1, antibody negative, other sorts of issues where their pancreas may not be working right, but 1,500 have a known cause. The distribution looks something like this, where you can see the green line is over 600 people in our registry with glucokinase mutations. And then HNF1 alpha comes in second. On the left are the neonatal diabetes. From that one case with Lily, we now have 143 cases just of the case in G11. But if you add them all up, it's over 200 cases of neonatal diabetes, which I have to tell you, in 2005, I'd never heard that there was such a thing. What also is interesting is I mentioned cascade testing. So if you look in the middle, that where it says GCK, and there's the two big bars, so we have 354 probands, but 623 altogether. So that means there's a lot of family members. That gives you a sense that it's a really reasonably benign. People go on to have children, and they do well, and they have large families. So typically finding a glucokinase mutation means you have a whole family. We had one family visit from Ohio last week, and to see a pediatric endocrinologist because the 7-year-old had high blood sugars and a glucokinase mutation. But I then said hello, and I said, well, who else has it? Well, mom had it, mom's sister had it, and mom's mom had it. So immediately we had 4 people with a glucokinase mutation that I love the pediatricians, but they didn't seem to take a family history. So what are the frustrations? Well, the genetic diagnosis sometimes makes no clinical impact. Sometimes people don't grasp the diagnosis. Sometimes the better results with non-insulin treatments may not work. There are programs around the country that are now doing this: Maryland, Mass General, Denver, Stanford. We want to put Miami on this list. There are multiple sequencing resources. So when people say, oh, I can't get genetic sequencing, we'll help you. And admittedly, it can be challenging. It used to be much more expensive. What was $5,000 is now $500. And so there is potential out of pocket or other sorts of ways to do it. I want to tell you about our Radiant grant. So we've been doing this now almost 7 years. It's a $5 million a year project funded by the NIDDK, and weirdly, they're still funding it. And it's us in Chicago, the group in Baylor, and Jeff Krischer at South Florida, and the group in Mass General are leading it with cores around the country. We're looking to identify new kinds of diabetes, discovery, smarter testing, accurate diagnosis. And sometimes it's the patients, people with diabetes, who contact us because the doctor said, I don't know what you have. So then they contact us. We try to get all this data, clinical, omics, laboratory, and stored samples, And then we're looking for people who have something that isn't defined very well. We also are, this is our status. There have been a couple of papers already, and a whole raft of papers are about to come out. We're interested in, I mentioned, monogenic type 1 diabetes. So people with, this young lady had type 1 thyroiditis, hemolytic anemia, and food allergies, and it turned out she had an interesting mutation in a macrophage-related gene, which is not surprising. That was surprising to cause type 1 diabetes. And this is the consortium that we're developing for people who are interested in this area. We have 14 centers. Toronto joined last week. And the idea is to answer questions that are not answerable without more folks. So if the n's are small, we're not going to be able to figure out what the phenotypes are. And I'll just end with this. So monogenic diabetes is an opportunity for precision medicine. It helps us understand what type 1 or type 2 might be. It helps us to understand, to ask the right questions. Why are whole families involved with one kind of diabetes or another? That's our registry. These are some of the folks. I'm proud of the rainbow nature of people from all over the world who have been able to work with us, and some of the funding agencies and the people themselves who have diabetes. Oh, and then I wanted to show you what I came away from. So thank you very much.
Thank you so much. I'm pinch hitting for Dr. Weiss, who had an urgent matter to which to attend. So thank you so much. I think precision medicine in all of our fields is so important. I actually thought even with SGLT2s, there's a lot of issues in endoscopy about periprocedural euglycemic decay, and I wonder how some of these things could even inform people at risk for that. I can't imagine glycemic control sleep at University of Chicago not bringing up something for Dr. Punjabi to ask, but I'll open it up for anyone to have any questions. Go ahead. Let me just see if I can.
That where you talked about this potassium ATP channel mutation. I was wondering about this subset of patients who will not respond to sulfonylureas because their channel is a little bit altered. I know the rationale for that might be low because it's very rare, and the subset would be even more rare. I'm just wondering, in the era of precision medicine, if there's any research effort to like modify the sulfonylurea molecule at all to like be able to treat those patients, or we just treat them with insulin.
Yeah, so we could talk for a couple hours about that. I think that the problem is the channel is broken in such a way that the sulfonylureas can bind, but the channel won't close, so it just stays open. And part of the problem is the developmental aspects. So by the time the child is born, it may be too late to do some of those things. We don't exactly know what's reversible. So Dr. Greeley, my colleague, has put together quite a few subjects who were started very early on and with sulfonylureas to see whether those most severe ones, whether anything improves in terms of psychoneurological function. And the answer is sort of a little bit. So if we could identify them prenatally, that would be helpful. And there are a small number where, so a parent survives and then has a child, it's dominant, so half the children will have the same thing. So then we can get in earlier. And people have asked us about gene therapy and whether that could be done. And the answer is yes, I think it could be done. But generally speaking, gene therapy right now is reserved for people with what the oncologists call a hard endpoint. So we're not quite there yet. A great question.
We have about a minute left.
Fantastic talk, inspirational. I'm a basic scientist, so my question will be related to what we do in the lab. So this is a unique opportunity to establish causal relationships between individual molecules and diabetes or even glucose metabolism. So is there any effort to get tissues from these patients and to study them in vitro later on?
So yes, so in the RADIANT study, where we don't know yet what the gene is, so part of that is PBMCs, for example. So for some of the PBMCs, we are imagining you could then de-differentiate them and make stem cell production and then whatever tissue you want eventually. In some of the cases where they're unknowns, then we're saying, okay, we do a muscle biopsy. There are some things to do. And otherwise, the idea is to give talks like this. And then if people have an idea, well, gee, that looks like something I'm doing, then, you know, we have basically a living biobank. So then we have the ability, what our registry allows us to do is to recontact. So we follow people over time, we try to stay in touch, and if there's something that happens or there's some additional interest in getting tissue, then we can contact them.
Well, we're at time. Thank you so much to Dr. Philipson for a wonderful talk. Just a reminder, CME and MOC are either in the chat if you're online or the QR code outside. And next week is our last grand rounds until the holiday break. So thank you guys.